seed viability in the genus Mimulus. Before I get started, I just want to take a quick second and acknowledge our postdoc, Elin O'Neill, who's just been super instrumental in this kind of work. So, as speciation biologists, we're all very interested in the evolution of reproductive isolation, how that accumulates through time. And in particular, I just want to draw your attention to this idea that hybrid seed viability is a super common reproductive isolating barrier in plants. And in fact, in the genus Mimulus, we know of at least 5 species so far that, when crossed in Bucatus, make these inviable, sad, shriveled seeds that just don't germinate. And so today I'm just going to focus on this one species, Mimulus decoris. And if you're a Mimulus aficionado and you've never heard of this species before, fret not, it's very newly introduced. So I'm going to introduce it, take a second to do that, talk about patterns of seed inviability, and then I'm going to jump back to the theory and talk about why we care about hybrid incompatibilities and this idea of parent of origin effects. I have some preliminary results on the genetic architecture of this hybrid incompatibility and some model results from that. And then, if there's time, I have a little bit of data to talk about the sort of intra-species variation of reproductive isolation. And I'd be happy to talk about that with other people later if they have thoughts. So Mimulus decoris is this really newly described species that was just sort of resurrected in 2014. It's relatively geographically restricted to the Cascades in Oregon, Washington, maybe into British Columbia. It's a diploid. It's very closely related to Mimulus Bucatus, and in fact lives in really close proximity to both annuals and perennials. And when it lives with perennials, they're root and root, sympatric, tangled mess. And it's very phenotypically similar, although there are some distinguishing characteristics. But one trait in which is very similar between these two species is that of seed production. So both Bucatus and decoris make viable, well-formed, plump seeds. But when you cross them, you end up with 2 very distinct seed phenotypes. When Bucatus is mom, you end up with these nice, plump, viable seeds. But when decoris is mom, you end up with these shriveled, wrinkled seed shells. And it turns out you can do a bunch of crosses, and there doesn't seem to be a lot of post-mating prezygotic barriers, so they all make about the same number of seeds. But if you look at the actual proportion which are viable, it's only in this decoris by Bucatus cross where you end up with strong seed inviability. And in fact, you can plate them out and see if they germinate, and they do not. So there's about 1% or less viable seed made in this cross. So you might look at this and just think like, oh, you don't even have seeds there. They're just not even fertilized. But it turns out there's sort of 2 lines of evidence that make us think that they are in fact fertilized seeds. Number one, they make a seed coat, which is a sort of direct consequence, or a consequence of fertilization. It only happens after that. And the second is that we've taken seeds which are really young, 5 days after pollination, and you can clear them and look at them under a microscope and see that they have an endosperm developing. And I realize this is probably super hard for you guys to see, but this little bitty circle here is a really young embryo that will eventually become the actual plant, and then all these little dots that are around it, those are the nuclei of the endosperm developing. So these are our parents. You can see they have nice embryos and they have an endosperm developing. And in the hybrids, you see both an embryo and endosperm. There's a little embryo. And yet, just 9 days later, this cross here, this decoris by Bucatus, is almost a completely empty seed. There's really not much going on inside of it. And as evolutionary biologists, we've sort of long been interested in post-zygotic isolation. It's sort of a puzzle because selection would never favor the direct evolution of unfit hybrids. So how do these barriers evolve in the first place? And Dobzhansky and Mueller, and to some extent Basin, came up with a really great model some time ago to explain this evolution. And I'm not going to belabor this point because we've heard about this many times today so far, but essentially the take-home is that these within population processes leading to the evolution of novel alleles in isolation, when they come back together and are found in a hybrid context, these newly derived alleles form novel genetic combinations which have never been tested before by evolution and result in the formation of inviable or sterile offspring. And yet we have a lot of questions remaining despite the fact that we've had this great model for a long time, and those are, what are the genes actually responsible for these incompatibilities, and why are they evolving within populations to begin with? Is it drift or is it selection? And if it is selection, is it driven by ecology or is it driven by something else like conflict? And today I'm just going to give you sort of a cliff notes on this idea of parental conflict and how that might contribute to reproductive isolation, particularly in Mimulus. So parental conflict is just this idea that moms and dads have different optima for their resource allocation. Moms are equally related to all of her offspring and thus will favor equal resource allocation among them. Dads, in contrast, in systems where there's multiple matings, are not equally related to all of the offspring in the litter or flesh, and thus should favor the selection for sort of greedy offspring. And it's important to note that greediness in this case is coming at the expense of other seeds in the pod or pups in the litter, also at the expense of mom. So moms and dads can easily enter into this coevolutionary arms race. And this is particularly true in plants because of this phenomenon called double fertilization. So when a plant ovule is fertilized, each pollen has two nuclei. One fertilizes the egg cell down here and eventually become the plant, and the other fertilizes this diploid central cell, and that becomes a triploid nutritive tissue. It's very similar to endosperm. It's called endosperm. It's very similar to placenta. And there's some sort of interesting things about this endosperm. Number one is that it has a weird genetic composition. It's two parts mom and one part dad. And in fact, we know that this ratio is really important for seed development because some really early crosses, you sort of mess around with this ratio by using polyploids. And it turns out when you have the wrong ratio, you really can't make seeds very well at all. And yet, many hybrid seed incompatibilities are actually from diploid crosses. And so that's really interesting. So it's not a problem of dosage effects. And in fact, there's been a lot of work on endosperm development, and it turns out a lot of genes in the endosperm are imprinted, whereby only one parental allele is expressed and the other is silenced. And it's a non-random parental allele. And it's been hypothesized that this imprinting in the endosperm is a direct sort of result to reduce, or a direct consequence to reduce parental conflict. And so the question really then becomes, do these within population processes that are potentially evolving to reduce conflict, are they involved in these intraspecific incompatibilities? So I've created an F1 backcrossing scheme to try and get at this. So I've made viable F1s, remember only from one direction of the cross, and I backcrossed them in both directions to both parents. And just to sort of step you through the model, when we're talking about parent of origin effects, it matters what your genotype is, but it really matters which allele you got from what parent. So in this case, we both have F1s that have the same genotype, but in one direction they got this M1 allele from mom and the P2 allele from dad, and that combination is the combination which makes aborted seeds. Okay, so just walk you through this model. If you take your F1, use it as mom, and you backcross it to chorus, you can inherit either the M1 or the M2 allele from mom, but you will always get the P1 allele from dad. None of those combinations are lethal, so all those seeds are fine. If you do the opposite, though, where you're using the F1 as a dad, you'll either get P1 or P2, and you will always get M1. And so in this case, 50% of your seeds are going to be aborted. This is just for a two-locus model. We get the opposite results when we do the backcross in the opposite direction. And so we can just put those data points up, so we have this sort of nice predictions for what we'd expect with a two-locus model. And next I'm going to show you another genetic model that could explain sort of an asymmetric cross, and that's a cytonuclear incompatibility. And the important point to remember here is that, or to see here, is that the only cross which differentiates these two predictions is this F1 bicutatus. And you don't really expect to see any aborted seeds here because these F1s have the cutatus cytoplasm, and then they're also 3/4 cutatus genetically. So what do the data actually look like here? And so what first take home probably is that they don't really fit with either super well. But what I really want to draw your attention to is that this cross really doesn't fit with the cytonuclear incompatibility. You really don't expect any seeds to be viable in that cross. And the other thing is that these data might not fit perfectly with a two-locus model, but it's in the right direction. So we can sort of fiddle with the number of maternal and paternally inherited alleles to try and get a prediction for how many alleles are contributing to this incompatibility. So I'm going to show you each model separately. This is the maternal model. This is just the predicted amount of inviable seeds according to the number of maternal loci, given that you already have the wrong paternal genotype. And here's where the data fall. So it looks like it's pretty close to sort of a two-locus model on the mom side. We do the same for the dad side. the data followed here, which is really similar to a 3-locus model. And I'll just say that I've also looked at F2s where you take an F1 and you self it. And this proportion of aborted seeds in that cross is pretty compatible with a 2-maternal, 3-paternal locus model. So the next thing we're doing is we're taking all of these 4 backcross categories and sequencing them, and we're going to look for segregation distortion, try and actually map the genes involved in this incompatibility. And so I think I have just a couple minutes left, and I want to just spend this time talking about this idea of heterogeneity in reproductive isolation. And so I sort of set this up, this talk up talking about like, what are the genes and like what are the evolutionary mechanisms, and those are super cool questions. But I think as speciation biologists, we sort of tend to think of like species as these homogeneous things, and yet as population biologists, we know that populations are super variable. And we've seen some great talks so far about variation in reproductive isolation, and I'm going to join the ranks on that one. So what I did was I took 20 populations of Mimulus decorus from throughout the range, and I crossed them reciprocally to 4 different populations of Cutatus. And here I'm just showing you the results of those crosses across both phenotypic space and geographic space. So the main take-homes here is there's a ton of variation, and you see that it's not really correlated with phenotypic space, so things that are more phenotypically similar are not crossers. But there seems to be maybe a mild correlation here with geography, in that it's this barrier is pretty restricted to the Central Cascade Mountains. And so one possible explanation for this is that all of these other light blue populations that are crossing really well are just co-occurring with Cutatus. They're really close by, and so that barrier is just dissolving through time. So luckily I have these 2 sympatric collections that I have where I have decorus and Cutatus. They're root and root sympatric, just a total mess. And I cross them, and I'm wrong. It turns out it has nothing to do with how close you are to a Cutatus population, because these species have a really strong crossing barrier, but you'll probably notice that it's in the wrong direction from what I just showed you before. So that's really wild. And I really just started thinking about this data like 3 weeks ago, so fresh on the mind. So just to wrap up, I've told you about this Mimulus decorus, which is a new species of perennial Mimulus. It's a really strong asymmetric crossing barrier with Mimulus Cutatus. The F1 seeds do appear to be fertilized, but they're just aborted really early on. The F1 backcrosses are consistent with a model of parent of origin effects, not a cytonuclear incompatibility. And it seems like the extent of the crossing barrier is just really complicated, and there's probably a lot of sort of within population processes that are important. But more on that later. Thanks. I have like 30 seconds for questions.
Me?
Yeah.
Do we know how the imprinting is actually inherited?
Yeah, so it's really complicated, not surprising, but it turns out that it's there's a little bit of like methylation that happens where like everything is methylated, and then the maternal side is preferentially